I'm Doug Robinson. I work at Novartis Pharmaceuticals. I've been here for the last 11 years. Before that, I worked at Bristol Myers Squibb for 5. I have a background in statistical genetics and really have been working on focusing and analyzing biomarker data. So this includes both genomic, proteomic, expression data. But this type of background has given me the ability to try and think about, well, how do we communicate with information, especially large amounts of data that we generate in our clinical trials. You know, the typical way of doing this with listings and tables just really breaks down and doesn't work well for clinical teams. So you have to really think about the way you visualize these data, and in that way, enhance your ability to communicate. So for the last 2 years, yes, I've been leading the scientific computing and consulting. I have to start with this disclaimer. Okay, so here's an outline of my talk. So I'll give you kind of a high-level background in the current state of affairs. I'll then focus on good graphical principles and what that means at Novartis, and share a really nice cheat sheet with you on graphical principles. And then the bulk of my talk will focus on R and Shiny at Novartis, and I'll show you some of the ways in which we're leveraging this new platform in order to collaborate with our clinical teams, and then wrap up with a conclusion. So to motivate the talk, why do things have to change? Well, so I'm not sure of the audience and the backgrounds of the audience, but I work in the pharmaceutical industry. Maybe, probably many of you do as well. And in general, pharmaceutical industry is really full of people who use SAS. There are SAS programmers, people who use SAS for decades. And unfortunately, SAS has not done a good job of keeping up and producing more dynamic displays of data. So in a sense, when we do our submissions to the health authority, we generate hundreds of static tables, listings, and figures, and regardless of value. So I had a clinician come up to me and they said, why are you producing all of this output? He said, we don't even look at 25, 30% of what you produce. That's really tragic, because those outputs don't come for free, right? They take a lot of time and effort for people to generate these outputs, hundreds of hours, and really it's a shame that they're not really used. And the problem is that oftentimes we try to cover all of the possibilities, so that when you write your analysis plan, the clinical teams put in absolutely everything that they want, everything that they could want, everything that they potentially want. And unfortunately, oftentimes the trials show unexpected results. These unexpected results were most likely not captured in their initial slate of tables, listings, and figures. So we have to go back and produce even more outputs to explore the data. And so in a sense here, what we need to do is think of an alternate way. Instead of using tables and listings, maybe we can convey even more information with graphics. So at Novartis, we have this idea of effective visual communication. And the idea is that you really need to build credibility with your collaborators, and in that way you earn their trust. In order to do this, you really have to understand the problem, right? The disease, the mechanism of action, the technology, especially the variability of how those data were produced. This is really important in biomarker data, when there's several different types of technology that are available to produce massive amounts of information. This is becoming even more acute with digital technology. People are wearing sensors in order to track their movements and take assessments on their patients. If you don't understand how those data are collected, then that's going to impact the way in which you assess those data. You really have to take the time to listen carefully to your collaborators, openly ask questions and clarify, really think about the problem. The whole goal of effective visual communication is to be able to convey complex information with ease. You want to be able to facilitate that insight, that understanding, and most importantly, facilitate the decision-making process. So at Novartis, we formed this Advanced Visual Analytics Initiative with the goal of enhancing the understanding with the use of what we call good graphical principles, and I'll get into that in the next couple of slides. We want to promote upfront graphical thinking and facilitate easy access to high quality graphics. This initiative was so successful that Novartis decided to formalize it with the creation of the Scientific Computing and Consulting team, which is the one that I lead. So as a shameless plug, and actually Naomi set me up perfectly for this, the mission of our team is to enable clinical teams to change how they interact with and interpret data through the development of dynamic and highly engaging web-based tools. I guess because I'm the speaker, I have the ability to say, yes, we have a position. We have a position in the Cambridge area. Currently, I'm actually located just south of Cambridge, Massachusetts. So it's a pleasure to be here. But our team, we have 2 people in Basel. We have 2 associates in Cambridge. And so we're looking to fill 1 headcount in Cambridge. And I have the link here below. What I'll do is, Naomi, is there a way to share the slides afterwards? Oh, Naomi, I think you're on mute. Oh, Naomi, I think you're on mute. Oh, unfortunately, she's on mute. That's all right. Well, anyway, I have the job here. If you go on, oh, yes, I can.
It said to use Control A, but that didn't work, but I see the unmute on the bottom left. Sorry about that. In any case, the whole recording will be shared if you give permission. They're recording the program, and we can certainly share the slides, and if you give permission, we'll share the whole program.
Okay. Yeah, I'm fine with that. Thank you so much, Naomi.
Fine.
So getting back to the topic at hand, I'd like to introduce what we call the graphic principles cheat sheet. So ironically, this slide doesn't follow the graphical principles that I promote in the cheat sheet, but essentially here, we give you the ability to download the cheat sheet in our open source GitHub repository. There's also an article about good graphical principles here. I give the link included as well. So what we've done is we've been able to share our ideas and thoughts with our community at Novartis about how to generate good quality graphics. What are the aspects you need to consider? What are some of the ways in order to facilitate comparisons? Think about the color emphasis, things you need to consider, and the legibility and clarity of the message that you're trying to convey. And so I highly recommend that you go and download this graphic cheat sheet for your own purposes. What we've done is we actually printed these out, laminated them, and gave them to every single statistician at Novartis. But I actually use this quite frequently, and I hope you find value in it. Now, I'm not going to go through all aspects of the cheat sheet, but I will go through a few. So essentially, the goal here is you really want to use the cheat sheet during the design and planning of your graphics analysis. So effective visual communications convey this complex information in an easily digestible manner. They facilitate insight and understanding and decision-making, as I said. So if at first you don't succeed, don't settle for just okay. In my mind, if you're going to take so much effort to do your analysis, you should take just as much effort to do your presentation of your analysis as well, because these impressions matter. There are several questions that you can ask yourself. Why am I making this graphic? What is it that I want to convey? Who are the audience that I want to convey this message to? What is important to them? Where are we actually going to make this graphic? Is it for a publication, or is it for a PowerPoint presentation, or what's the venue in which this graphic is going to be displayed? These types of aspects actually change in the way you may want to use and generate that graphic. And when you think about the data that you're going to use, carefully consider the question that you're going to ask. Once you know the question, you want to strive for simplicity to maximize the value of your data. This is a nice aspect of the graphical cheat sheet where it talks about the different types of graphical information and the different ways to convey it. There's evolution, part to whole, magnitude. I'm not going to go through each one of these, but I'd like you to encourage you to take the time to review the front and back of the cheat sheet and encourage you to explore the information that we have available. So at Novartis, we not only encourage the cheat sheet, but we actually have what we call a graph gallery to make it as easy as possible for associates to select good graphics. The graph gallery is a curated list of graphics with simple data, with sample data and code. Currently, it has 130 sample graphs to view and fit your project needs. Here, what's nice is that these are graphics that are generated in SAS and R are the two main softwares in which we use to generate these graphs, but we also have graphs in MATLAB as well, and Monolix. These types of plots, what's nice is that we have sample data and the code so that you can download it, see the data formats, and generate the plots for your own graphical needs if that's appropriate for your purpose. And so there's several different options with color, some of them in black and white, especially if you're going to do a completed study report for submission. And all of these reflect the good graphical principles as was shared in the graphical principle cheat sheet. What I want to stress here is that good graphical principles, although they apply to static output, they also apply to dynamic graphs as well. And so here in scientific computing and consulting, we try to make it as easy as possible as well to share information about our dynamic visualizations. So what we have is an app hub. So it's kind of like a shiny gallery for internal Novartis information. We have a software called REM, which is an R library, which helps us with reproducibility. Sometimes when you make a graphic, what happens is that if you change the version of R or don't have the correct dependencies, that graphic won't render properly and you'll probably get some errors. REM is a really nice package. If you don't know about it, I highly recommend you use it. It's basically based on PackRat. It's the next generation PackRat version. And what it does is it actually helps you maintain the version of R, the packages and their dependencies to reproduce that environment to generate that graphic. Or your analysis, by the way. We'd like to generate several different templates to make it easy for our developers to produce applications to explore their clinical trial data. We actually have one called Shiny Mobile, which is an app template for mobile technology. So if you have an app, this is a way to have it render on your iPhones. We've generated standard packages for templates, such as the dashboard layout. On the next slide, I'll go through the templates that we have. And on the screen here is a new way to modularize your code. We call it tidy modules. And here, this is a link to our open source GitHub page where we actually have the source code, documentation, examples that explain this new way to modularize your code. It's actually based on an object-oriented approach using an R6 class information. And one of the benefits is that it facilitates module-to-module communication. And what that means is that if you're building a Shiny app, you want to be able to select points in one of the displays. Now, once you select those points, how do you see those people in another visualization? How do you pass that information from one module to another? Using Shiny modules, it's actually really quite difficult to pass this information, but using tidy modules, we actually developed a way to facilitate this module-to-module communication, and it really solves this problem in an elegant manner. So I highly recommend that you look at the open source GitHub page and download this information and see how it could work for your app development needs. As I said, SCC, Scientific Computing and Consulting, has developed several Shiny templates for our associates to really enable their development of applications. We have 3 different types of templates. First of all, there's the simple page, which is just the all-in-one. You have your options here and the graphic in the middle. There's the nav bar page, which actually has tabs to navigate from one plot to another. And the third one is the dashboard page, which is based on the BS4 Dash package. So we are very fortunate to have the developer of the BS4 Dash on the SCC team. And so what he's done is he's actually made a version that's compliant with the Novartis color schemes and formatting, so that we have our own kind of internal BS4 Dash template that people can use to develop their applications. These templates have 3 different features. There's authentication, so you can actually sign in and access different types of repositories. Once you sign into the application, the app has that ability to sign into different servers using your login information. It has authorization, so you can actually restrict who has access to that information. This is really important, especially if you're dealing with sensitive trial results. And it also has usage statistics. So if people start logging on, you can actually track who logs on, the frequency with which they log on, the number of unique users, that length of time that they spend on each page of the app. And so this helps you understand like which components of your application are actually benefiting your audience more than others. There's several different benefits of these templates. They're a professional looking application. There's unified headers and footers. The colors are compliant with Novartis brand scheme. And what's nice is that since folks are using these templates, they actually have a real consistent look and feel, so that people can actually learn how to use one app and quickly evolve to another in a quick and rapid manner. So one of the main ways in which we support clinical projects is through the development of these Shiny applications. And so I'm going to kind of go through some of the types of visualizations that we create. We kind of think of the clinical projects as the ideal breeding ground for new ideas. We actually work with the clinical teams and see what resonates with them. Then we actually see what may be generalized and used across these different programs. Now, I will say that sometimes you could go in and you say, okay, this is what you want, and it just doesn't fit that team's thought at the moment. So what we try to do is we try to go in and try to be very open with the way in which we collaborate with these teams, and we try to see like what actually works for their specific purposes. So here, this is a stacked bar chart where you have, say, different treatment groups, and these are the different time points, and you see how the categories change over time within each one of the treatment groups. We do a lot of longitudinal data analysis at the individual level. We can also do means and confidence intervals. This is using ggplot2. We do, in our oncology studies, we do a lot of waterfall plots. Box plots are another one that we do. Here's a nice one. This is called a swim lane chart. So here you have the duration of response. Out here, we have the the different patients on the y-axis. And here we have the different events, say whether it's adverse events or information that is important to the trial is overlaid onto these swim lane charts. So you can see the distribution of the lengths of time for each patient from one treatment group to another. Here's a very common Kaplan-Meier curve, very popular. What I like about the Kaplan-Meier curve, it's for looking at time to event information, but this, which is often associated with oncology, but oftentimes this is used across the board. For instance, in cardiovascular, we have the time to myocardial infarction or the time to exacerbation in respiratory. So this actual plot is used quite a bit. We've actually been able to overlay the subgroup and subpopulation functionality here, so then people can see how the Kaplan-Meier curves change for different subgroups in their studies. This is one that's called a lasagna plot. Actually, I just learned that name. So this one is what happens here is you can look at how patients change in a categorical variable that's assessed over time. We're actually using this in the COVID-19 trials where people are assessed in the WHO 9-point scale. And what we want to see is that what was their scale at their score at baseline, and how does that change for each patient over the study. In this way, then you can compare your treatment group to a control group to see if those people actually change to a better state more quickly than the other competing treatment. Here's one where we're trying to use animation. Sometimes we have endpoints that are composite endpoints that are made of multiple component parts. In this way, what we have here is that we can actually look at, for instance, the ACR score in arthritis. Here you have different components that make up the ACR measure, and you want to see, well, how does each component impact the overall ACR score? And here what we can do is we can actually dissect that ACR score and see how each individual component changes within each treatment arm of the study. We also focus on non-clinical projects. So here is what we call a sunburst metric. So this is, we call this the analytics resource visualization tool. And this helps us understand who we have in the department, where they're located, what group they're a part of. And it's based on a custom built database and visualization tool. We also have the ability to work with our statistical methods group in assessing the probability of success. Here, they've developed a very sophisticated assessment to look at different projects and assess their probability of actually going into submission and being successfully at the health authorities. This complex methodology is very difficult to use on its own, but what we've done is built a very nice user-friendly interface that provides, a really nice way to open up this complex statistical methodology to a broad clinical team base. After we've been able to look at and see what resonates with our clinical teams, we then go back and say, well, I wonder if this could actually be useful to other clinical teams as well. And in that way, what we've done is we've generalized a lot of these tools, one of which is this Safety Explorer. We have a patient profiler, patient listing generator, a time to event, and then a subgroup identification toolbox. Here, what we've done is we have the live link to the live version of the app, which is available for download, the complete app documentation. And one of the things that we found is that there's different ways to convey this information to our population of associates. Some people like to read the documentation and read about all of the functionality. But what we've also done is we've created mini video libraries to also demonstrate the functionality in short video clips as well. We actually have an internal GitLab source repository and issue tracker as well. So we recognize that, and in some cases we try to generalize this code, but there still might be issues in certain projects. So what we do is we have the ability for certain clinical teams to come contact us and say, hey, actually this isn't working for us. And then we can actually go in and dissect what those issues are. So one of the key generalized tools that we have at Novartis is basically a safety explorer suite. One we recognize is that safety is a common challenge across the board. This is based on the Safety Explorer suite from Roe Inc. It's incorporated. It's a company down in North Carolina. This was actually part of the ASA DIA working group where they actually focused on safety exploration. And so one of our members in Novartis was actually part of that working group and was able to bring in that software in collaboration with Roe. The AE Safety Explorer is nice because what it does, and this package, I highly recommend you go here and you can download it yourself and use it in your studies. Here, what we have is we have each treatment group, the AE rates. This is done in tabular form, as well as dot plot and forest plot. Here, these are organized by system organ class, and if you click on the plus sign, it actually has them organized by preferred term under that system organ class. It has kind of a nice searching feature, so if you type head into it, you can actually get headache, tension headache, and any type of item that's related to head. And it helps you really navigate the different pages of the AE listing that you have. Here's another one that's called the AE timelines. This one is nice because what it shows is for each patient, it shows their AE history ordered by time point of the first adverse event. Here, the AEs are color coordinated by severity, and here black circles represent severe adverse events. You can mouse over the start of each one of these, and what this does is this actually brings up what that adverse event was, when it started, when it ended, and the severity score for each one for each patient. And so that's really helpful. We find that our safety monitors and our clinical teams like to dive in and actually focus in on certain patients in the study. But we have many folks that are still in love with SAS. And what we've done here is we've actually integrated the two. So I'm actually gonna end this and bring up a live demo of this tool. Here, what we've done is we've actually been able to implement a logistic regression model. And many of you who know SAS may recognize the proc logistic function call here. What we've done is we've wrapped this into a Shiny module where the person specifies the treatment, the response, and then they can specify the subgroup. So we can choose that, gender as well. And what we can do is then submit that to our SAS server. So this is on a Shiny server. And what we do is in the back end, we actually do a function call to the SAS server. The SAS server responds and we can actually pull in the list here, as well as the log information. So our associates can actually review and explore the log and see if there's any errors in there. But what's nice is using the ODS output, we can actually bring in the output data sets and then render them using the power of R. So this is actually using a ggplot2 for the forest plot. And so here we have the overall effect, and then we have it for each one of the subgroups. So the age group categories here, BMI categories here, and then gender here. And then we have that same information in tabular form, and then the treatment by category or subgroup interaction score as well. So this is really kind of nice because this allows us to blend the worlds of SAS and R into one kind of nice interface. And so what's nice is that here, you can imagine that this app is for logistic regression. We're going to see how the clinical teams like this and the analysts, and if they do, you can imagine building out a suite of tools. For instance, you can leverage Proc Mixed, Proc GenMod, other types of procedures that you do. And because we recognize that one tool may not serve everyone's purpose, here now we can actually leverage the power of the SaaS platform and bring it in and leverage the good graphical principles of R to blend the two and actually open up this new resource to folks who want to explore their data in a more dynamic fashion using the SaaS. So, right. So I was talking to Naomi last week about the presentation, and she brought up one of the neat examples that I displayed way back in 2018 at the New Jersey chapter of the ASA. And so back then, this was a very cool example of visualization. So I credit the following folks at Novartis for doing it. Here, one of our compounds focuses on alleviating joint swelling. So patients are measured at approximately 70 different joint positions and are assessed whether they're swelling or not. The data are essentially position, and then 1 and 0 for yes, is it swelled or not. Data are taken at baseline, week 8, week 16. So for one patient, you have 210 measurements. And that means that for a huge study that we have in this area, there's actually 3.1 gigs of data with thousands and thousands of rows of data. So that makes for a really complex way, and how are you going to visualize that information? Well, this team came up with a very cool way to look at and explore that information. And they did it using this, what we called kind of a humanoid figure here. So each data point is accurately placed on a human figure. The dark colors represent whether there's swelling or not. And so then what's nice is you can easily change the treatment group, you can change the patients, you can click through the patients in the study. And interestingly here, for this one patient, that's actually a 7-page listing. And yet you can see all of that information in one very easily digestible plot. So this is kind of a neat way in which people are rethinking the way they view this information. This was also extended to skin scores. So we have scaling, thickening, PASI score. And so you can see how using the same kind of humanoid, you can have darker indicating worse conditioning, where lighter is better. And you can actually see how the patient may change at different time points in that study. So that was back in 2018. So the question is, well, where is that visualization now? So the following, what happened was the Novartis hired an external vendor to bring in and really try to expand upon this original idea. Idea was to use much more realistic graphics to show these scores. Joint scores are shown as red areas with deeper shades of red indicating higher scores. And you remember on that previous slide, we had skin scores as well. So on the skin, it's actually shown as blotches indicating disease severity. So this is what it looks like right now. And so this shows just how amazing graphical thinking and really graphic arts can actually change the way people look at their information. So using a patient's score, right? Remember on that previous slide, we had joint tenderness and joint scores here. That's the same type of information that's used on this figure as well, with joint tenderness and joint swelling, using a more realistic feature. We also have scores showing the disease, and this can change over time. I actually have a promotional video. I'm not sure I can. I can certainly show it. Is that allowed, Naomi?
Certainly.
Okay. So hopefully you guys like this. So there is no sound, but this is kind of the promotional video for the compound in interest. So the compound is called secukinumab. And here you see some of the screenshots that I showed on that previous slide. Here we had the joint scores and skin scores. And here we see how those joint scores and joint swelling is actually put onto the graphical representation. So this app is actually dynamic, where you can actually see how this patient is reflected over time. And you see how the blotches become less red, showing that the pain and swelling has gone down. Here we're now looking at the differences in the skin scores, and you can see how it clears over time. And I think this one's kind of cool. So they use that digitization, and they have that ability to put that onto the figure itself. So that's kind of neat. Shows you where things could be going in the future. Okay. So here, in conclusion, so Novartis is on a real journey here to harness the potential of its data. So again, here's our job posting, right? I'll take the opportunity to show that again. SAS is still strongly rooted in our formal submission process, but by using Shiny apps and using dynamic displays of our information, teams have recognized the limitations of static outputs and really have a strong desire for that flexibility and interactivity. However, there's still some analytical capabilities of SAS that are still really quite desirable. And you saw this ability to try and reach out to SAS on the server side to bring in that information and be able to perform those analyses while still providing high quality graphics with the resultant data sets. We continue to focus on good graphical principles as the key to our success. This is critical for both static and dynamic graphics. So the graphic principles cheat sheet is here. Highly encourage you to download it. Shiny's dynamic interactivity is a powerful tool in collaboration with clinicians. We really want to reduce down the number of static tables, listings, and figures, and really promote this concept of learning from your data rather than asking for anything and everything up front. And so when you have teams that are really engaged, senior management support and statistical colleagues that say they're having fun building these tools, then that's really the ultimate win-win. So here is my contact information, and thank you so much. I really enjoyed presenting to you today.
And thank you so much for presenting. I'd say everybody give a round of applause, but you won't hear it unless we all unmute ourselves, which I don't recommend. For questions and answers, I don't see a Q&A on the bottom here, so I guess the best thing would be to use the chat function to ask questions. Doug, I noticed that one person did ask a question. How do you determine the gender and how did you determine the gender and ethical representation of the human there? And anybody else who has questions, why don't you type them in the chat and I'll ask Doug to review the chat.
So I'm trying to share. Can people see me now?
I can.
Okay.
Yep.
Great. So to the person about the humanoid figure, so I have to admit that I was not involved in that type of visualization. The team that actually did the kind of the stick figure kind of skeleton figure before, those were within the analytics department. All of that other stuff that I showed you with the realistic humanoid, that was done by an external vendor. So I'm actually not sure how they assessed that.
Other questions? Again, use the chat function to ask a question. I see
Let's
lots of thank
see.
you and compliments, but I don't see questions. Anybody?
Indian more formal than stick figure. Let's see. How to determine the gender. Okay. Uh, Thank you. Thank you. It was a pleasure. How big is the community of analysts at Novartis that are using the cheat sheet around the world? So, you know, we've actually been presenting this cheat sheet now for 2 years, and we're really trying to open it up to a much broader community. And every time we can actually give the opportunity, we try to present it. When at the ASA New Jersey conference, we actually printed it out for everyone there. Actually, last fall, I gave a talk at the ASA FDA industry workshop in Washington D.C. Put the GitHub link up there as well. I actually don't know how many times it's been downloaded, but you know, I kind of feel like it's kind of a nice compact way that kind of makes you stop and think a little bit about what it is you're trying to present, and has some really good helpful hints on best ways to do graphical representations. I find sometimes we try to do too much with our graphics. Actually, too much actually starts to detract from the message at the end of the day. So you really want to strive for that simplicity and really focus on the message. So it takes time to do a good graphic, and I highly recommend you taking that time to produce that. See, how are you handling publishers to include individual data points on graphics? How are you handling the request by publishers to include individual data points on graphics? Individual data points. I mean, you mean sharing the clinical data? I'm not exactly sure what you mean there. Maybe you can type more in the chat window. Um, is the data used on all drugs? So what we're doing here on my team is actually, it's really kind of nice where we're situated in the department in Novartis. We actually have a sight line into all phases of development. So we actually work with the early development as well as the full development where we do our submissions, as well as the late phase GMA, Global Medical Affairs, the phase 4 clinical trials as well. So our tools, we try to generalize the tools as much as we can so that they work across the different phases of development. And what's nice is, if you notice, we focused on adverse events. So adverse events is one of these things that is universal regardless of whether it's respiratory, neuroscience, ophthalmology. Adverse events is always a concern. And so we felt that if we could focus on adverse events, that's something that actually could resonate across the portfolio, which is why we tried to work on it first off. What we're now doing is we're now kind of focusing in on different therapeutic areas like oncology. And so there, you know, in certain therapeutic areas, there's a lot of therapeutic area specific graphics. So we're actually trying to selectively choose which graphics makes the most sense for each of those DUs, and really try to develop tools that maybe work well for that therapeutic area, but may have potential across different therapeutic areas. I gave that example of the Kaplan-Meier curve, where you have different types of word problems, time to event type of challenges, actually resonating in different therapeutic areas. So that was why we went for that one. Can you talk more about how long it took to move to Shiny Apps, how your team did it? That's a great question. So, you know, what's happened is it's still an evolution at Novartis, right? And I have to say that one of the nice things is that we have a Shiny server that sits on top of our clinical database, and I think that that's actually a game changer. What that does is when we have our Shiny apps, we actually have access to the clinical information. So there's certain other companies where the Shiny server isn't attached to the clinical information. So then it's much more challenging to develop these tools. What we've done is we've actually selected key projects that have high visibility in the department and in the company to develop these tools for. And we call these kind of hypercare projects, where we invest a lot of our resources into developing these tools. This allows us to have much greater exposure across that therapeutic area and across the department in the development of these tools. And that's actually broadened our ability to get the word out and advertise our existence. Right now, we're we're actually victims of our own success, which is why we're trying to hire. It's kind of a nice place to be, where we have actually having to say no to some of these teams coming to us, because we just don't have the resource to do it. Right now, what we're trying to do is generalize a lot of the functionality that we're building. And in that way, if you do that in a consistent manner, then you can be able to potentially build these applications much more rapidly and be able to help support a much broader community with these tools. So we're trying to see like what tools can be used across the different therapeutic areas, as well as, you know, writing them and generalizing them enough so that we can pick them up and use them in our subsequent studies. How do you, let's see. How do you come up with new visualizations techniques using the cheat sheet? So I have to say that, you know, for those of you who use R, it's an amazing community of people out there, and the number of different ways people are coming up with to visualize their data, it's really remarkable. One of the things that we do is we try to have an eye towards what's being released out there, and really look at the different libraries and packages that are being released, and try to kind of explore what's available. You know, we go to meetups like this and see how people are presenting their information, and see if that works, and then what resonates with folks. We also talk to our clinical teams, because the clinical teams are reading, you know, very different journal articles, and they said, oh, by the way, I read this article and they had this visualization in it, and I'm wondering if you could do something like that for us. And so then they share the article with us, and we try to understand, you know, what was used to produce that. So really the source of inspiration comes from a wide variety of sources. It's not one source of information that we get our ideas from, but it's kind of you know, it's one of these things that's evolving, continually evolving. So you actually have to make the time and effort to keep up with it. There's a lot out there. I have to say that, you know, even in R, we haven't explored the full capabilities of R, and there's just so much flexibility out there. One of the other things that we're trying to do is expand beyond R into JavaScript. So maybe many of you know about the D3 library, D3.js. Yeah, D3 it's just incredible. The variety and the complexity of the graphics that they have there is just amazing. I think that you know, there's really no shortage of really creative and innovative people out there that we can actually learn from. Is there different criteria used for different drugs? Well, I think there's different endpoints for different drugs. So, you know, in oncology, it's a lot of time to event. In respiratory, there's continuous measures, so they're using a lot of mixed effects models. So it really, the graphics really stem from the questions that are asked, right? And so once you have a really good question, that really drives the type of visualization that you generate. So, you know, I do think that there's a lot of things that resonate from one therapeutic area to another. One of the plots is the Sankey chart. which shows how patients flow from one category to another. We're actually trying to use that for several different categorical data analysis as well. So you have the different categories being measured over time. Yeah, seeing how patients flow from one category to another. Yeah, it really is based on the question, the type of data that you have, and then what resonates with that clinical team. Those are really the criteria. Um, are there analysts at are the analysts at Novartis centralized, distributed in functional areas, or both? The answer is both. The Novartis is a really large company that has the ability to look at and explore many different therapeutic areas and different diseases within those therapeutic areas. In the team that I'm a part of, so I'm part of the clinical development and analytics community, and essentially there we have all of the statisticians and pharmacometricians that are really facing the drug development pipeline. So there you have the early phase, the late phase, so where you do the submissions, and then the global medical affairs, as well as pharmacometrics, which look at drug-drug interaction, dosing regimens, different types of ways that drug is administered, food effect studies, population pharmacokinetics. And so our team, so each one of those groups is like therapeutic area aligned. So that's like within each one of the areas, you have a statistical team associated with that. But then you have the team that I'm a part of, which is called Advanced Methodology and Data Science. That's actually sits kind of along the side and is not necessarily therapeutic area aligned. So it actually has the ability to help out different therapeutic areas as well as different phases of that development. So there, it's kind of like a centralized team that's able to help out across. So it's kind of a mixture of both. And I mean, I don't know which way is right. I think each way has its pros and cons. I think size is a challenge, but then communication, right? Getting the word out and making sure we share the information from one therapeutic area to another. Because one of the things we want to be able to do is if someone develops a tool that's really nice in cardiovascular, we want to make sure that that's actually available and potentially enhanced for use in neuroscience or oncology. So we really want to try to leverage the power of our people and really encourage them to explore different ways to visualize the data. So I guess one of the things I wanted to cover was that the analysts coming out of school now are actually they're not being taught SAS anymore, and so they're coming out fluent in R. And so, and many of them have already developed shiny apps even before they graduate. And so they're coming in with ideas, and what we want to do is really promote that innovative thinking and really try to leverage the talents of the whole department. And then enable the department to develop even better tools. So really trying to share that knowledge. We have 2 monthly sessions where people can actually share their information and share their project work with one another. We have one that's much more technical where you can peek under the hood to see, well, how did you do that? How are you communicating from one module to another one? What new packages are out there, and how did you use that visualization? How was that experience with that clinical team? So we try to have these forums to promote that community and sharing. Why R Shiny and not Dask, since Python is more common these days than R? Great question. I think here there's not one tool that works well, and I think you know, it's interesting. I think we chose R because it's a statistical shop, right? And so the statisticians that we have and pharmacometricians come out fluent in R, as I said, and what we wanted to do was build tools that then they could use, and they could you know, modify on their own, and maybe tailor it to their specific purposes. I think what I've seen in the industry, at least in my purview, is that Python is growing. Um, from what I've seen, I mean, R seems to be a little bit more rooted, but Python is growing. And it really, I have to say, it depends on the question that you're asking. No one tool is going to solve all your problems. And one of the things that's coming up right now at Novartis is that people want to start looking at imaging data, right? And they want to do machine learning or artificial intelligence. And I have to tell you, R is good, Python is better, right? Hands down. And so actually what we're thinking about doing is actually using increasing these types of visualizations for Python as well, because that's where the questions are, right? I think you want to use the tool that's best apt to answer those questions. For our image analysis, it happens to be Python. You know, for machine learning, it's Python as well. So we want to try to increase our skill set in Python as well. And maybe at some point it will overtake R. But I do see that you know, we want to be open to the different technologies out there. And right now with Shiny, then using Flask and Python, you know, I think we're going to evolve in that direction too. The other one is JavaScript. Actually, JavaScript blows the doors off of both of them for interface design, HTML interface design in Java and JavaScript programming. You can't beat it. And so actually we're actually thinking about, for our position, people who know Java and JavaScript to help with our front end to really develop very powerful and aesthetically pleasing interfaces using the JavaScript libraries. I mean, you know, there's a whole host of things that we can learn. So we're trying not to focus in on one. And are open to expansion. Are you able to see measurable results from its implementation? It's really all encompassing with some very sophisticated technique. Is it available in other languages? Able to see. So John, are you able to see measurable results from its implementation? Very sophisticated techniques too. Is it available in other languages? So John, I'm not sure if you're referring to the figure or the humanoid, or you're talking about
below. Below, he said he meant the cheat sheet.
Oh, the cheat sheet. Oh, okay. Is it available in other languages? Right now, no. That's a great question. That's a great suggestion. Other languages. That's a fantastic suggestion. We haven't done that, but I'm going to bring that back and actually get it translated into different languages. Thank you so much for the suggestion. That's actually really helpful. I like that. Have you been able to address reproducibility in case the analysis generated through the Shiny app needs to be submitted to the FDA without reprogramming in static? Great question. So one of the things we're actually, we haven't done any submission to the health authority using Shiny, but what we're doing is using that REM, that reproducible environment package that I told you about. What that does is, again, focuses on the version, the packages, and the dependencies. So then you can actually reproduce that environment, okay? So that if you increase the R version on your server, the REM actually goes back to the original version in which you created your app, and then focuses and is able to reproduce that environment. And I think that's the first step. The other thing we're doing is using Docker containers, right? So then here you can actually have a containerized Docker container around your app, and have the R version, the packages, the dependencies. You also have the code and the data locked in a container, which can help you do that reproducibility. You know, although we haven't done any thing with the FDA right now, it's actually an active source of discussion, and people are seeing the potential there. And actually, the FDA is really quite open to it. I've really been impressed with the FDA's ability to actually embrace R and Shiny. They actually have come to the R in Pharma, which is a nice conference that's held at Harvard University in the summers. It's going to be virtual this year. But we actually had folks from the FDA come to that, and they're saying, yeah, we're doing Shiny all the time. So they're very open to it. How we're actually going to navigate that, I think, is going to be an evolution and a process. But you know, I think the global goal is to actually stop these tremendous paper submissions, right? And actually have an app that actually has the information and allows you to explore the information you have. I mean, that's the ultimate goal. And we're on a journey to get there. And hopefully we'll get there someday soon. Given that SAS is so embedded, was there an evaluation of SAS visual analytics? How did R Shiny become the preferred? Great question. So SAS is really quite embedded, but we actually don't have the SAS visual analytics solution at Novartis. And R is free, and people came in knowing it. And so when we actually got the Shiny server as part of one of the upgrades of our infrastructure, people just gravitated towards it. You know, honestly, it was really organic. How we just started working with it, playing with it. And the interesting thing is we started getting positive feedback from our clinical teams. So, you know, when the clinical team started liking it, it went up to senior management started liking it, and it just basically started to snowball from there. Want individual values included in graphics to represent the N. Ah, I see. Right. Yeah, so I don't have an answer to that. What individual values included in the graphics? You know, let's see. I mean, I think here, you know, Plotly allows you to get those individual values, but I don't know how you do that in a graphical representation for a static submission. Yeah, I don't know the answer to that. Forgive me. Okay, can you guys still hear me? I guess someone couldn't hear me. Can you hear me okay? Okay. Have you tried out Julia? How do you feel it compares with the R ecosystem of tools? So the answer is no. I haven't used Julia, but I'm open to it. And honestly, I think all of us have to be open and flexible to the evolution of how these softwares are going, because there's a lot out there. And again, no one tool can do the solution. And it's possible that Julia can do things that R can't. And I think we should be open to that. So I'll thanks for that question. As in the cheat sheet, thanks. I assume there are hundreds of people. All right. Exactly. Yeah. I mean, so Novartis is a huge community. We've shared it with our colleagues at Roche, which is another huge community. We're sharing it with all of you. It's an open GitHub page. Share it with your friends. I mean, we print it out and give it to our colleagues. You know, give it to your friends, your family, read it to your kids. I'm joking, but I mean, honestly, I mean, we put it on the open source GitHub page with that link for the very purpose for it to be freely shared. And honestly, I mean, if you want to print it out and hand it out to your entire company, please do. Honestly, I mean, if it helps you, we want to promote the good graphical thinking, and really get away from these massive listings, and summary tables. Not to say that, you know, there are some tables that are really good out there. I mean, those should stay, and there's nothing wrong with a good table, but I think visually people can get a lot of information out of a real high quality graphic, and you know, we're trying to explore that space right now. When you look at a completed study report, and submissions, I mean, it's much more tables and listings than figures. So I think there's a huge opportunity there. SAS and SPSS have traditionally worked well with huge data sets that do not fit in memory, and a lot of analysis could be pushed into a database server and even across a cluster for computers. For our users, are there good solutions for this kind of problems where the data is huge and does not fit into memory of a single server? Which solutions did you like the most? This is a great question, and actually this is one that we're struggling with. You know, the way SAS reads in data is it has them, it stores it in flat files and then reads in parts at a time, so it never has the full data set in memory. And with that, you can do massive analyses, and it has a way of handling that in the back end. And I actually think that that's one of the significant strengths of SAS. You know, when I was at Bristol Myers Squibb, I was doing mixed effects models on gene expression arrays. So we were doing longitudinal microarray studies in our immunology portfolio with several different dose groups, and we had, you know, hundreds of patients at several different time points. And so then we had something like 400 or 500 microarrays across the study. And yeah, I did that analysis in SAS doing mixed effects models for the different groups, and I mean, it worked great. I couldn't do that in R, honestly. I couldn't. And so here, what we do is at Novartis, we're trying to explore different back-end databases to have the ability to read in that data and then be able to access just what we need. There's a couple of different issues there. One of the things is do you need all of the data that you have? When you look at SAS clinical data sets, you know, oftentimes the clinical data has like 100 columns in it. And there's a lot of redundancy and a lot of information in there. But for your graphic, you might only need 10 of those columns. So one of the things that we do is we actually trim down the data to just the columns that we need, and that significantly reduces down the size of those data sets. And like for laboratory data, what we try to do is we try to talk to the clinical teams and say, well, are there key lab parameters that you're interested in? And so that we don't have to read in all of them, and maybe just focus on those that the clinical teams are interested in. That can also reduce down the data set size. Now, once you get a data set, so let's say you have, even if you do that, there are times when you have, you know, these massive, these mega trials in cardiovascular. You know, we had one out there that was like, gosh, it was like 12,000 patients, right? And so let's say you're looking at creatinine levels, which is a common measurement that's measured multiple times for those patients and what have you. And we were looking at, I remember we were just looking at baseline values, and we just wanted a scatter plot of it. And even trying to render the scatter plot blew the memory, just of the baseline values. And so then you think, well, are there different ways of showing that data, right? And so maybe instead of a scatter plot, you can use these tile plots, which actually have like these hexagonal tile plots, which then have different colors associated with it, so you can see kind of the depth in terms of color, and that actually doesn't use as much memory there. So that's kind of the solution that we went to for that. So I think there are different ways that you have to handle it with R, but you're right. I mean, SAS actually got that one really, really well, and we continue to struggle with it. What about Excel? Visual Basic for stat and graphics add-ins can handle millions of rows now. Do your apps run in Windows and iOS? iOS is more secure. So we actually run it on a Linux server. So everything is on a Linux server right now. Yeah. How best to contact you for follow-up questions? So I gave you my contact information. Maybe I'll put it up one more time. Uh, uh, uh,
We can post it. Oh, okay. He has it here.
so there it is one more time. So it's just me, Douglas.Robinson@Novartis.com. Email me. You know, please reach out. I'm really enjoying the discussion. All right, I'm going to close this so I can read the messages again. Okay, so here's one. When migrating databases, do you use an ETL tool? So that is out of my area of expertise. We have an IT department that handles that. We just use the data itself. So I apologize. I don't have an answer to that. Suggestion. So Alam says, please consider using databases in your work. Postgres is freely available and has a great set of R integrated tools. You know, Alam, maybe you and I should talk because you know, I really like this idea of using databases, especially relational databases. You know, and I think SAS data sets actually are really big because I don't think it's the best way to store store information. Like, it has a lot of overhead associated with SAS data sets. I think there are much better ways to store information, and relational databases are really really quite nice. And so here, I have to say there seems to be a movement and an evolution towards having databases of the data that we have. And one of the questions that we have here is that the data that we have is getting larger, right? So as I mentioned, there's a lot of digital data, and people are beginning to look at the images as well. So here's an example. You know, immunohistochemistry. You know, when we get those data, if you don't know what that is, immunohistochemistry, what you do is you take the cells and you stain them for a protein expression, and the darker the color is, the greater the protein concentration is. And what's nice is that you get the pictures of the cells. You can take a microscope picture, and then you can see where that protein is localized. Is it in the membrane? Is it in the cytoplasm? Or is it in the nucleus? Or is it in the mitochondria? Right? That, where the protein is localized, might have give you better information about how that information may be used, and how that protein is being distributed within the cells. And so the way it has typically come into clinical trials and research is that a highly trained pathologist takes a look at this and gives it a score based on what they see in the information, and they're highly trained, and they can do this. One of the suggestions out there is, well, maybe if we use image analysis, we could potentially look at a wider swath of that slide image, as well as maybe have a more reproducible way of assessing that protein expression, and get maybe even more information out of it. And so, you know, what I see is people are collecting this information, and now because the tools have evolved, we're actually thinking about how can we use that information even more, and maybe get even more insights out of it. And this was information we threw out not that long ago. So I'm saying this because you know, the data are changing, and the volume of the information is changing, and so we have to evolve as well. And I think having a database to store this information and to pull it up quickly, given its size, is something that something we have to do. Is very easy and useful. RStudio. DB Plier, yeah, it makes it very easy and doable. Excellent suggestion. Excellent. Yeah, DB Plier for database. Fantastic. Which features, capabilities, if existed, would help you and your team be a lot more productive when working on your projects, or even enable you all answer time-consuming questions that cannot be answered today? So that's a great question. Really, this is fantastic. So I'll answer this, but I see that we're running short on time. I think for me, it all comes down to education. You know, I guess one of the things that I see is that in the statistics department, we're really not trained in computer coding or coding style and coding quality. I think here people should learn how to code well, and then that makes it more reusable. So there's been a lot of times where our team does a lot of consulting with others, and people are doing really nice work, but because they don't code as well, their code is very difficult to follow, and it's difficult to generalize. I think if we could have maybe an emphasis on good coding quality, right? Coding quality principles, if you will, then the code might be able to be transferable, and we could leverage and harness the people's in the organization. A coding cheat sheet. You're exactly right. I mean, you know, Hadley Wickham has done a real nice job in thinking about how to code well, and I think it's a process. And for computer scientists, this is second nature. But for those of us in the statistical community, I think we code to get the job done. We don't really think about the quality of the code. But like, does it work? Great, it's done. Let's I have another project to do, right? But I think if people actually took the time to document their code, to really think about good coding quality and aspects about that, being neat and organized about how you code, writing up documentation around it, that could really ease the reproducibility of that code, and could make it easy for other people to take and use in their projects. And I think that at the core could then be used to expand the libraries that people are generating, and could actually help us generate apps and tools much more quickly. So with that, I just want to say again, it's been an absolute pleasure talking with you all. I hope you got a lot out of the presentation. I hope it was enjoyable to you. Naomi, honestly, it's been a while. It's a pleasure to talk with you again. It's a pleasure to see you, and I really want to say sincere thanks for inviting me to talk at this forum. It's been great. Please come contact me, and everyone, please stay safe out there. Thank
I
you.
want to say what a pleasure it was seeing you again and hearing this talk. Everybody here, judging by the number of questions and the response, it was highly successful. I thank you so much for coming. I thank all of you for attending. And again, our next speaker will be George Camos from Lisbon. We've been wanting him for a long time, but couldn't fly him in from Lisbon. He will be speaking either the end of July or mid-August. We avoid early August because that's when the joint statistical meetings are. The joint statistical meetings will be virtual this year. Normally, I have to pay for airfare and a hotel for 5 nights. This year, there's just registration if anybody's interested. They're the first week in August, so we would not be meeting then. And George will be talking about lessons from the pandemic, state of his lessons from the pandemic. Again, thank you all for attending. Thank you, Doug. And thank you, Mahabu, for hosting us. We really appreciate it. That's it.
Thanks, everyone. Bye-bye.
Thank you.
Oh, he's a very good speaker.
Yeah.